we can solve the mystery beyond doubt of who this skeleton actually is. None other than Cleopatra's sister, Arsinoe, Egyptian rebel, Queen of Egypt.
For over 2,000 years, the world was taught Cleopatra came from an unbroken Macedonian Greek bloodline. Now, new DNA results from a mysterious skeleton at Taposiris Magna hint at a lineage no historian predicted. The markers don't match the royal profile we've repeated for centuries. They point somewhere far more controversial. If this is Cleopatra's true ancestry, it rewrites everything. So, who was she really? This revelation changes everything. The discovery that changed everything. Taposiris Magna is an ancient temple complex located about 30 miles west of Alexandria, Egypt. For years, archaeologists have been excavating this site, hoping to find answers about one of history's greatest mysteries. What happened to Cleopatra after her death? Where was she buried? And what secrets did the last pharaoh of Egypt take to her grave? The temple itself was dedicated to Osiris, the ancient Egyptian god of the afterlife and rebirth. It was built during the Ptolemaic period, the same dynasty that Cleopatra belonged to. The location has always been significant because historical records suggest that Cleopatra had strong connections to this particular temple. Some researchers believe she may have even funded renovations and additions to the complex during her reign. In 2022, excavation teams working at the site made an unexpected discovery. Deep within the temple grounds, beneath layers of sand and rubble, they found a burial chamber. Inside this chamber lay the skeletal remains of a single individual. The burial showed clear signs of high status and importance. The skeleton was positioned in a way that indicated careful preparation and respect. The surrounding burial goods included fragments of gold jewelry, ceremonial objects, and decorative items that would have been expensive during ancient times. These are not the kinds of items buried with common people. This was someone who held significant power or influence in ancient Egyptian society. What made this discovery particularly interesting was the location. The burial chamber was situated in a part of the temple complex that would have been reserved for individuals with royal connections. During Cleopatra's time, access to these sacred spaces was strictly controlled. Only those with direct ties to the royal family or the highest levels of the priesthood would have been buried in such a place. Archaeologists began standard analysis procedures immediately. They examined the bones to determine basic information about the individual. The skeleton appeared to be from an adult who lived during the 1st century BC. This time frame aligns perfectly with Cleopatra's reign, which lasted from 51 to 30 BC. The burial artifacts provided additional clues. Stone fragments near the burial site contained partial inscriptions mentioning the royal court. While the inscriptions were damaged and incomplete, they clearly referenced individuals who served in positions of authority during the Ptolemaic dynasty. Everything about this burial suggested a direct connection to Cleopatra's inner circle. Traditional archaeological methods can tell us a lot about ancient remains. We can estimate age at death. We can identify old injuries or diseases. We can analyze burial practices and artifacts. But there are limits to what these methods can reveal. We cannot know for certain who someone was related to or what their actual family connections were based on bones and objects alone. That is where modern science comes in. Researchers working at the site proposed something that would have been impossible just a few decades ago. They wanted to extract DNA from the skeleton. Genetic analysis of ancient remains has become increasingly sophisticated in recent years. Scientists can now pull genetic information from bones that are thousands of years old and use that information to trace family relationships, ethnic backgrounds, and ancestral origins. The decision to pursue DNA analysis was significant. Ancient DNA extraction is expensive and technically challenging. The genetic material in old bones is degraded and fragmented. Contamination from modern sources is a constant risk. Success is never guaranteed, but the potential rewards were enormous. The bone samples were carefully collected and sent to specialized laboratories equipped to handle ancient genetic material. The waiting period began. It would take months of painstaking laboratory work to determine if usable DNA could be recovered from the remains. But the real shock came when scientists decided to extract DNA from the ancient bones. Cleopatra's official bloodline. The story of Cleopatra's ancestry begins over 200 years before she was born. In 323 BC, Alexander the Great died unexpectedly in Babylon. He was only 32 years old and had conquered most of the known world, but he left no clear heir to his massive empire. What followed was chaos. Alexander's generals fought each other for control of different territories. One of these generals was Ptolemy, the son of Lagus. Ptolemy was smart and ambitious. He moved quickly to seize control of Egypt, one of the richest and most strategically important provinces in Alexander's former empire. Ptolemy declared himself pharaoh in 305 BC. This was the beginning of the Ptolemaic dynasty. For the next 3 centuries, his descendants would rule Egypt. They were Greek in origin, speaking Greek as their primary language and maintaining Greek cultural traditions. But they also adopted Egyptian customs and presented themselves as legitimate pharaohs to the Egyptian people. The Ptolemies had one very unusual family practice. They married their siblings. Brother married sister, generation after generation. This was not a Greek custom. It was an Egyptian royal tradition that the Ptolemies adopted to strengthen their claim to the throne. The ancient Egyptian pharaohs had practiced sibling marriage for thousands of years. The Ptolemies wanted to appear as true Egyptian rulers, so they followed this custom. This practice of sibling marriage meant that the Ptolemaic bloodline remained remarkably pure. There was very little genetic diversity introduced into the family over 300 years. The same Greek Macedonian genes passed from one generation to the next with minimal outside influence. At least that is what all the historical records indicated. Cleopatra VII, the famous Cleopatra we know from history, was born in 69 BC. She was the daughter of Ptolemy XII. Historical sources tell us that her father was of pure Ptolemaic blood, descended directly from Ptolemy I through centuries of careful sibling marriages. But there is a problem with Cleopatra's family tree. We do not know who her mother was. Some ancient sources suggest her mother was Cleopatra V, who was also the wife of Ptolemy XII. If this is true, then Cleopatra's mother was also her father's sister, continuing the family tradition. This would mean Cleopatra was of pure Greek Macedonian ancestry, with no genetic contribution from outside the Ptolemaic line. Other historians believe Cleopatra's mother might have been a concubine or a lesser wife whose name was never recorded. Royal men in ancient Egypt often had multiple partners. Not all of these relationships were documented in official records. If Cleopatra's mother was someone other than Cleopatra V, then her ancestry becomes much more uncertain. For over 2,000 years, most scholars assumed Cleopatra was entirely Greek. Ancient Roman sources described her as Greek. Renaissance paintings showed her with European features. Modern textbooks presented her as a Macedonian queen, ruling a foreign land. The idea of Cleopatra as purely Greek became deeply embedded in how we understand ancient history. This assumption shaped everything about how Cleopatra was portrayed and remembered. Her intelligence was attributed to Greek education. Her political skills were seen as Greek cunning. Her famous beauty was described in European terms. The possibility that she might have had ancestry from other backgrounds was rarely considered seriously. The Ptolemaic dynasty kept detailed records of royal marriages and births. These genealogies were important for establishing legitimate claims to the throne. But these records also had gaps and inconsistencies. Sometimes wives were not mentioned. Sometimes children appeared without clear parentage documentation. The further back in the dynasty you look, the more confusing the family tree becomes. What we know for certain is this: the Ptolemies claimed to be of pure Greek Macedonian blood. They emphasized their connection to Alexander the Great and the glory of Greek civilization. They maintained Greek as the court language and surrounded themselves with Greek advisors and generals. Cleopatra herself was reportedly the first Ptolemaic ruler who bothered to learn the Egyptian language. But claiming a bloodline and actually having that bloodline are two different things. Royal families throughout history have hidden inconvenient truths about ancestry, yet the DNA evidence was about to expose a secret the Ptolemies had hidden for generations: the DNA extraction and analysis. Extracting DNA from ancient human remains is one of the most challenging tasks in modern archaeology. Unlike fresh tissue samples used in medical laboratories, ancient bones have been exposed to environmental conditions for hundreds or thousands of years. Heat, moisture, bacteria, and chemical processes in soil all work to break down genetic material over time. DNA is a fragile molecule. It begins degrading immediately after death. In most environments, usable DNA disappears completely within a few hundred years. Egypt's hot, dry climate actually helps preserve ancient remains better than wetter environments. But even under ideal conditions, DNA from 2,000-year-old bones is severely damaged and fragmented. The research team at Taposiris Magna knew they had a difficult task ahead. They needed to extract whatever genetic material might still exist in the skeleton and then piece together enough information to draw meaningful conclusions. This would require expertise from multiple scientific disciplines and access to the most advanced laboratory equipment available. The first step was selecting which bones to sample. Not all parts of a skeleton preserve DNA equally well. The interior of dense bones offers the best protection for genetic material. Teeth are particularly valuable because the hard enamel shell protects the softer interior tissue. The petrous bone, a dense part of the skull near the inner ear, is considered the gold standard for ancient DNA extraction. Samples were carefully removed from the skeleton using sterile tools in a controlled environment. Contamination is a major concern in ancient DNA work. Modern human DNA is everywhere. A single sneeze or touch from an ungloved hand can introduce contemporary genetic material that will overwhelm the ancient fragments researchers are trying to study. Every precaution must be taken to prevent contamination. The bone samples were shipped to 3 separate laboratories that specialize in ancient DNA analysis. Using multiple independent labs is standard practice in this field. It provides verification and helps identify any contamination or errors that might occur in a single facility. The 3 labs were located in different countries, each with its own team of geneticists and specialized equipment. Inside these laboratories, scientists began the painstaking process of extraction. The bone samples were ground into fine powder. Chemical solutions were used to dissolve away everything except the DNA molecules. What remained was filtered and purified multiple times. The goal was to isolate whatever tiny fragments of ancient genetic code might still exist. The amount of DNA recovered from ancient samples is incredibly small. Scientists often work with just a few picograms of genetic material, that is, a few trillionths of a gram. These microscopic quantities must be amplified using a process called polymerase chain reaction, or PCR, which creates millions of copies of the DNA fragments so they can be studied. But amplification creates its own problems. Any contamination gets amplified along with the authentic ancient DNA. This is why authentication is so critical. Scientists use multiple techniques to verify that the DNA they are studying actually came from the ancient individual and not from a modern source. One authentication method involves looking at damage patterns. Ancient DNA has characteristic types of damage that accumulate over centuries. Modern DNA does not show these patterns. By analyzing the specific ways the genetic code has degraded, scientists can confirm they are working with genuinely ancient material. The 3 laboratories worked independently for several months. Each team used slightly different extraction and analysis methods. This independence is important because it means the results are not influenced by a single lab's techniques or potential biases. If all 3 labs reach similar conclusions, confidence in the findings increases dramatically. As the work progressed, researchers focused on specific sections of the genetic code. They looked at mitochondrial DNA, which passes from mother to child and can reveal maternal ancestry going back many generations. They also examined chromosomal DNA, which provides information about both parents and can identify ethnic origins and geographic ancestry. The analysis involved comparing the ancient DNA sequences to vast databases of genetic information from modern and ancient populations around the world. These comparisons can reveal which populations the individual was most closely related to. They can identify ancestry from specific geographic regions. They can even detect mixing between different ancestral groups. By late 2022, all 3 laboratories had completed their initial analysis. The data had been checked and rechecked. Authentication tests confirmed the DNA was genuinely ancient and not contaminated with modern genetic material. The results from all 3 labs showed remarkable agreement. The research teams prepared to announce their findings. What they had discovered would challenge fundamental assumptions about Cleopatra's ancestry. When the results came back, the laboratory fell completely silent. The shocking results. The DNA results appeared on computer screens in 3 different laboratories on the same day. Scientists stared at the data, checking and rechecking their calculations. Something was clearly different from what everyone had expected. The genetic profile did not match the historical narrative that had been accepted for over 2,000 years. The mitochondrial DNA analysis revealed the first surprise. Mitochondrial DNA is passed directly from mother to child with very little change across generations. It serves as a genetic signature that can trace maternal lineage back through time. When researchers compared the ancient DNA from Taposiris Magna to databases of known populations, they found genetic markers that were not typical of Greek or Macedonian ancestry. The skeleton carried mitochondrial haplogroups, commonly found in North African and Sub-Saharan African populations. These are genetic signatures that originate from maternal lineages in regions south of the Mediterranean. They are distinct from the haplogroups typical of Greek, Macedonian, or other European populations. The presence of these markers indicated African ancestry on the maternal side. This finding directly contradicted the established understanding of Ptolemaic bloodlines. If this individual was connected to Cleopatra's family, as the burial location and artifacts strongly suggested, then the pure Greek-Macedonian ancestry claim was incorrect. Somewhere in the family tree there was African heritage that had never been documented in historical records. The chromosomal DNA analysis provided additional information. Unlike mitochondrial DNA, which only traces maternal lines, chromosomal DNA comes from both parents and can reveal a more complete picture of ancestry. The results showed a mixed genetic profile. There were indeed Greek and Eastern Mediterranean genetic markers present, confirming some connection to the Ptolemaic dynasty's claimed origins. But there were also significant contributions from African ancestral populations. Genetic analysis can estimate what percentage of someone's ancestry comes from different geographic regions. The Taposiris Magna skeleton showed approximately 30 to 40% African ancestry. This is not a small amount. This is a substantial genetic contribution that would have come from relatively recent ancestors, not from ancient mixing that occurred thousands of years before. The 3 laboratories reached nearly identical conclusions despite using different analysis methods. This convergence of results strengthened confidence in the findings. When independent teams analyzing the same samples reach the same conclusions, it indicates the results are reliable and not due to laboratory error or contamination. But who exactly was this person? The DNA could tell researchers about ancestry, but it could not provide a name or specific identity. What the burial context suggested was that this individual had close connections to the royal family. The location, the burial goods, and the time period all pointed to someone within Cleopatra's immediate circle. One possibility is that this was a half-sibling of Cleopatra. Royal families often had children from different mothers. If Ptolemy XII had children with a woman of African descent, those children would be part of the royal family, even if they were not in the direct line of succession. They would have been buried with appropriate honors in locations connected to the dynasty. Another possibility is that this was Cleopatra's own mother or grandmother. The maternal lineage of Cleopatra has always been uncertain. If her mother or grandmother had African ancestry, this would explain both the genetic results and the historical silence about these women. Royal families sometimes deliberately obscured inconvenient truths about ancestry. The implications were staggering. For centuries, Cleopatra has been portrayed as Greek. Every description, every artistic representation, every historical analysis has been based on the assumption of pure Macedonian heritage. If Cleopatra herself had significant African ancestry through her maternal line, then our entire understanding of who she was needs to be reconsidered. The DNA results also raised questions about how common this mixing might have been in the Ptolemaic dynasty. If one member of the family had African ancestry, were there others? Did the supposedly pure Greek bloodline actually include genetic contributions from Egyptian, North African, and Sub-Saharan African populations throughout the dynasty's 300-year history? Ancient royal families were skilled at controlling their public image. They wrote the official histories. They commissioned the artwork. They determined which information was recorded and which was suppressed. The Ptolemies presented themselves as Greek rulers maintaining Greek culture in Egypt. but the genetic evidence suggested a more complicated reality. The research teams prepared their findings for publication in peer-reviewed scientific journals. They knew their results would be controversial. They anticipated pushback from historians who had built careers on established narratives about Cleopatra and the Ptolemaic dynasty. But understanding what this meant required looking at secrets the ancient world desperately tried to bury. Rewriting ancient history. The DNA evidence from Taposiris Magna forces us to reconsider what we think we know about the Ptolemaic dynasty. For 300 years, these rulers presented themselves as purely Greek. They spoke Greek at court. They worshipped Greek gods alongside Egyptian ones. They maintained Greek cultural traditions and surrounded themselves with Greek advisors. But genetic evidence suggests the reality was far more complex. Ancient Egypt was not an isolated society. It sat at the crossroads of multiple civilizations. Trade routes connected Egypt to sub-Saharan Africa, the Mediterranean world, and the Middle East. People moved along these routes constantly. Merchants, soldiers, slaves, and travelers brought diverse populations into contact with each other. Genetic mixing was inevitable in such an environment. The Ptolemies ruled Egypt for 3 centuries. During that time, they needed to maintain their grip on power in a country where they were foreign rulers. The native Egyptian population vastly outnumbered the Greek ruling class. Smart rulers would have formed alliances with powerful Egyptian families. Marriage was one of the most effective ways to create these alliances. Royal men throughout history have had relationships with women who were never officially acknowledged. Concubines, secondary wives, and unofficial partners were common in ancient royal courts. Children born from these relationships were sometimes recognized and sometimes hidden, depending on political circumstances. The official genealogies recorded in stone inscriptions and papyrus documents may not tell the complete story. The silence in historical records about Cleopatra's mother is particularly significant. We know who her father was. We know about her siblings, but her mother remains a mystery. Some sources claim it was Cleopatra V, her father's sister. Other sources are vague or contradictory. This lack of clarity might not be accidental. If Cleopatra's mother had African ancestry, there would have been strong political reasons to obscure this fact. The Ptolemies based their legitimacy on their connection to Alexander the Great and Greek civilization. Admitting to mixed ancestry could have weakened their claim to rule. It could have been used by rivals to question their right to the throne. Controlling the narrative about royal bloodlines was a matter of political survival. The practice of sibling marriage in the Ptolemaic dynasty may have been partly about maintaining genetic purity, but it was also about concentrating power and wealth within a small family group. However, this practice created serious biological problems. Inbreeding leads to genetic defects and health issues. Some researchers believe the Ptolemies may have quietly introduced outside genetic material to counteract these problems, while publicly maintaining the fiction of pure bloodlines. Ancient Egyptian society had complex views about race and ethnicity. The Egyptians did not categorize people the same way modern societies do. What mattered more was cultural identity, language, and adherence to Egyptian religious practices. A person could be considered Egyptian regardless of their genetic ancestry if they adopted Egyptian ways. The Ptolemies understood this and presented themselves as legitimate Egyptian pharaohs, even though they were foreign rulers. Cleopatra herself demonstrated sophisticated understanding of Egyptian culture. She was the first Ptolemaic ruler to learn the Egyptian language. She participated in Egyptian religious ceremonies and presented herself in Egyptian artistic styles. She connected herself to the goddess Isis and adopted Egyptian royal symbolism. If she had African ancestry through her maternal line, this might explain her deeper connection to Egyptian identity compared to her predecessors. The DNA evidence also challenges how we think about ancient identity more broadly. Modern concepts of race and ethnicity did not exist in the ancient world. People identified themselves by city, kingdom, culture, and language rather than by genetic ancestry. The obsession with racial categories is a more recent historical development. Ancient people would have found our modern classifications strange and irrelevant. What the Taposiris Magna skeleton reveals is that the ancient Mediterranean world was genetically diverse. People from different backgrounds mixed together. Royal families were not isolated from these patterns. Even dynasties that claim pure bloodlines likely had more complex ancestries than their official records admitted. For Cleopatra specifically, the possibility of African ancestry adds new dimensions to how we understand her life and reign. Her political alliances, her relationship with Egypt, and how she was perceived by both Egyptians and Romans may all need to be reconsidered in light of this evidence. She was not simply a Greek queen ruling a foreign land. She may have had genuine ancestral connections to the African continent and its people. The genetic evidence does not diminish Cleopatra's accomplishments or historical significance. If anything, it makes her story more interesting and more human. She was a product of a complex, multicultural world where identities were fluid and bloodlines were more mixed than official histories acknowledged. The legacy and ongoing mysteries. The DNA findings from Taposiris Magna represent a turning point in how archaeologists and historians approach ancient remains. For decades, researchers relied primarily on written records, artistic representations, and physical artifacts to understand the past. But these sources can be incomplete, biased, or deliberately misleading. Genetic analysis provides a new type of evidence that cannot be altered or erased by ancient propagandists. More research teams are now pursuing DNA analysis of ancient Egyptian remains. Museums around the world hold collections of mummies and skeletons that have never been genetically tested. Each of these individuals could potentially provide new insights into the ancestry and relationships of ancient populations. Technology continues to improve, making it possible to extract usable DNA from increasingly degraded samples. The Ptolemaic royal mummies that still exist are obvious candidates for future DNA testing. Several members of the dynasty have been identified in museum collections. Genetic analysis of these individuals could confirm or refute the findings from Taposiris Magna. It could reveal patterns of ancestry throughout the dynasty and show whether mixed heritage was common or exceptional among Ptolemaic rulers. However, genetic testing of royal remains raises ethical and political questions. Who has the right to authorize such testing? Should ancient rulers be subjected to analysis they never consented to? How should results be interpreted and presented, especially when they challenge deeply held cultural narratives? These questions do not have easy answers, and different countries and institutions have different policies. The search for Cleopatra's actual tomb continues. Despite decades of excavation at multiple sites, her burial place has never been definitively identified. Ancient sources suggest she was buried alongside Mark Antony, her Roman lover and political ally. But the location of this tomb remains one of archaeology's greatest unsolved mysteries. Dr. Kathleen Martinez, who led the excavations at Taposiris Magna, continues to believe the tomb may be located at or near the temple complex. The discovery of the skeleton with African ancestry has only strengthened her conviction that this site holds important secrets about Cleopatra and her family. Further excavations are planned, though they proceed slowly due to funding limitations and the complexity of working in such a historically sensitive location. Other researchers focus on different potential burial sites. Some believe Cleopatra's tomb is underwater, submerged by earthquakes and rising sea levels that have dramatically altered the coastline near Alexandria over 2,000 years. Underwater archaeology in the region has revealed remarkable structures and artifacts, but nothing definitively connected to Cleopatra's burial has been found. The DNA evidence from Taposiris Magna also highlights how much we still do not know about ancient population movements and mixing. North Africa in the Ptolemaic period was a meeting point of multiple civilizations. Greek settlers, native Egyptians, Nubians from the south, Phoenician traders, Jewish communities, and many other groups all lived in relatively close proximity. Understanding how these populations interacted genetically requires much more data than currently exists. Modern debates about Cleopatra's ethnicity often miss the point of what the ancient world was actually like. People in the 1st century BC did not think about race and ancestry the way we do today. They cared about citizenship, culture, language, and political allegiance. Cleopatra was Egyptian by virtue of being pharaoh, regardless of her genetic background. She was Greek by virtue of speaking Greek and participating in Greek cultural traditions. These identities were not mutually exclusive. What matters most about the DNA findings is not that they allow us to categorize Cleopatra into modern racial classifications. What matters is that they reveal the limitations of relying solely on historical documents written by people with their own agendas. Ancient writers had reasons to emphasize certain aspects of royal identity while downplaying others. Genetic evidence provides an independent line of inquiry that can confirm or challenge what written sources tell us. The skeleton from Taposiris Magna reminds us that history is always being rewritten. New discoveries force us to reconsider old assumptions. Technologies that did not exist a generation ago now allow us to ask questions that were previously unanswerable. Our understanding of the ancient world will continue to evolve as more evidence comes to light. Cleopatra remains one of the most fascinating figures in ancient history. The new genetic evidence adds complexity to her story rather than simplifying it. She lived in a diverse, interconnected world where identities were more fluid than we often imagine. Her legacy extends beyond questions of ancestry to encompass her political brilliance, cultural sophistication, and the dramatic circumstances of her life and death. The mysteries surrounding Cleopatra and her bloodline are far from solved, but we are asking better questions now than we were before. And sometimes, knowing what questions to ask is the most important discovery of all. If you enjoyed this video, like and subscribe, and also click the next video shown on your screen.